Hello everyone, my name is Stephanie, and the topic that I've picked for my presentation and research paper is embryonic stem cell research. So what are embryonic stem cells? Well, they are basically cells that are taken from embryos while they are still in the early stages of their development. They are produced in vitro, which means that they are taken out of the mother's uterus after fertilization and placed in a petri dish of sorts where they will develop like normal embryos. Eventually, after they have hit a certain stage in their development, scientists are able to extract their cells and use them for stem cell therapy. So, why are they used? There are a lot of ways that these cells can be used, but the most important way is through embryonic stem cell therapy. So, during the early stages of development, humans do not have specialized cells like red blood cells or skin cells. They are what is known as pluripotent, which essentially means that they can become any cell that we need them to be. Patients with Parkinson's disease, for instance, which is a degenerative disease, eventually with time lose a lot of control over their basic motor functions, and these cells can replace the ones that are causing the disease, which helps the patient. It allows them to live a healthier and longer and more fulfilling life. These cells are brand new and they're healthy, and they multiply constantly to help the patient for the rest of their lives. So why are they important? In this slide, I have a diagram which shows a basic step-by-step process of how the cells are harvested and then transferred to another human. So obviously, to make an embryo, a sperm and egg cell are needed. They come together, and once the egg cell is fertilized, something called a blastocyte or blastocyst occurs. It is basically a small clump of cells that are undifferentiated, which means that they do not have a specific cell function. This usually occurs in the third or fourth week after fertilization, and this is when the cells are harvestable and pluripotent. This is the stage also where they are usually put in the dish and then are able to develop into the specific cells and be transferred to people for therapy. In this next slide, I also have a small diagram of the cultivation of the actual cell and what they look like during their phases. So in the very beginning, when it's still in vitro, it's just one small egg cell, and then when it enters the blastocyte stage, it becomes a small clump of cells. And in the next portion, we can see inside where the stem cells are actually sitting. And once we take those out, we can convert them into basically whatever cells are needed to help the patient. So with this amazing research comes a lot of issues. I wanted to talk about the actual controversy surrounding the issue and what it means. So the issue with the cells is that the embryo is a human, and although the mother gives consent, it is a form of abortion and it is considered killing another human. Because what happens is when the cells are harvested, it kills the embryo. There is no way for the cells to be harvested and keep the embryo alive because it is such a small clump of cells that so many missing it cannot support itself in that way. And so people have been very angry concerning this research, and the government has been ignoring the public for the most part for a long time. A lot of scientists and researchers have been looking to get laws and bills passed that would allow them to do research a specific way, and the people have had quite the opinion on it, but the government has been deciding on these laws and bills themselves and ignoring the people, which has been infringing on their First Amendment right to voice their opinions and to take part in democracy, which has also caused even more controversy. And it is also not fair for someone else to decide whether a person lives or dies, especially when this embryo will become a human being and they cannot decide for themselves whether they want to be a science experiment or they want to live. Otherwise, it is a great discovery and it has helped many people live better lives. In this next slide, I have my quote that I picked for this presentation. I spent a lot of time looking for a quote that I wanted to put on here. This one has to do with the First Amendment. This woman, Catherine, she comes from the National Coalition Against Censorship Alliance, which is a group of about 50 or so organizations, and they specialize in helping spread the word and knowledge about equality and civil rights, where the First Amendment is concerned. And they speak a lot about issues that infringe upon the First Amendment, which is what she was writing about concerning the embryonic stem cells in this specific piece of writing that I pulled her quote from. Her alliance really looks to promote and defend the First Amendment's freedom of expression and thought, and I thought it was a very good read because it not only concerned my topic, but it also concerned the amendment that I wanted to go with it. So it was very interesting to see what her thoughts were on it. And this next slide is my thesis statement. In my research paper, I do go a lot more into depth about the topic and I imply my stance on the issue. I think that my thesis statement really explains the gist of the topic pretty well because it is a very new discovery. I think it's been around for only 20 or so years, and in the science and medical field, that is very, very new just because a lot of trial and error and hypothesizing has to go into these types of things, and it takes time, especially when cells are concerned, considering DNA was only discovered about 20 years ago, and we're still figuring it out. And these cells are amazing, and they can do so much stuff that we didn't know was possible and can help so many more people than we thought was possible. As for my research paper itself, as a whole, I'm almost done with it. I'm about 3/4 of the way done, and I'm continuing to work on it slowly and adding more elements in and trying to make it all presentable. In this last slide, I do have my source evaluation. This source that I picked specifically, I'm not going to use in my paper just because I did not get it from the database. I got it from Google, but it's a research paper. It's a very large research paper. It's about 15 to 20 pages, and it is entirely on embryonic stem cell research and whether it is unethical or why it is ethical. And it also talks about adult stem cells as well, which are what we have. They are stem cells basically, but they already have specialized functions, which is why they cannot be used the way that embryonic ones can, because embryonic stem cells are pluripotent, whereas ours are not. Ours are in a fixed state and they cannot be changed in any way. And this research paper was very interesting to read. It gave me a lot of ideas and topics to include in my own research paper, and I was able to highlight it a lot and refer back to it whenever I had a question about something. I also believed that the person who wrote it was very viable because I saw on their research paper that they wrote this for a medical academy, so they must have a very good sense of understanding of the medical field and the research that goes in, just because they have probably had to do a lot of research themselves. And so I thought it was a very interesting paper, and it was very cool to read and see what kind of information I could get out of it. And that is all. Thank you so much for listening, and I hope I was able to teach you something new today.